Hello everyone, welcome to Infinity IAS Academy. Today we are going to learn about the most used terms in the recent times, that is RT-PCR and RAT. I'm sure most of you would have gone through one of these two testings because of COVID-19. First of all, we should look at RT-PCR and then we will look into RAT. So what is RT-PCR? RT-PCR is reverse transcriptase polymerase chain reaction. It is combined with another technique called real-time reverse transcriptase polymerase chain reaction. What does it do? We shall start with what you know. So we all know we went through testing where either a nose swab or a throat swab is taken, and from there viral RNA is extracted. Why viral RNA is extracted from there? When COVID SARS-CoV-2 virus infects you, it basically contaminates your throat or under your nose. So the swab is taken where virus has something called as genome. Genome is there in every living species, including in human beings. So does virus. So that genome is extracted, and SARS-CoV-2 has something called as RNA. It is a type of a genome, and this RNA is extracted through the swabs from nose and throat. This RNA looks something like this. It is a single strand RNA. It has to be converted into a double-stranded DNA. How do we do that? So we use something called as reverse transcriptase. It's an enzyme. It is used to convert RNA to DNA. So the given RNA, using reverse transcription and the enzyme reverse transcriptase, it is converted into double-stranded DNA. This DNA is then amplified using a technique called PCR, which is polymerase chain reaction. And what do we use here? We use polymerase. First, we use reverse transcriptase to convert RNA to DNA. And to amplify DNA, we use something called as polymerase. After the DNA amplification is done, this DNA is kept into a machine called real-time machine. This real-time PCR, while it is amplifying the DNA, it also attaches a fluorescent molecule to the amplifying DNA. It can also be called as fluoropores. These molecules are attached to the DNA that is being amplified. Because it is a fluorescent molecule, we can detect it at a later time point. So the DNA is taken, real-time machine is used, and this amplification is detected using fluorescent molecules or fluoropores. So this is basically a simple version of RT-PCR along with real-time. Now, next, we shall discuss about rapid antigen testing. I'm sure most of you must have heard about rapid antigen testing, where you must have got kits to your home and you must have tested at home. So it's basically a point of care. It's basically a POC, point of care diagnosis. That means the place where the testing happens, that's exactly where the results are also seen. One other point of care testing kit, which is very similar to this, is pregnancy test. This also works mostly on the same principles, where you take either saliva or a swab from your nose or your throat, and then you use it on a testing kit, which looks something like this. Before going on to the testing kit, we shall look at the principles as to how RAT is used. So RAT basically uses a principle called antigen-antibody principle, where antigen is a foreign body that attacks you, such as virus, bacteria, fungus, etc. And antibody is something that is produced by your own immune system against the antigen that is invading your body. So Say suppose antigen looks something like this. It's basically a protein. It is from foreign. It is not your own body protein. It is a foreign protein that is coming into your body. And antibody is produced by your own immune system. This antibody recognizes the antigen, binds to it. So this technique is used in rat testing, where you have a kit and you take your saliva, you put your saliva into it, and then you see a line that forms if you are positive. If you are not positive, there is no line. However, there is a little bit more complexity to it. I will try and explain you in bits and pieces. There are two lines, as you can see. One is a test line. The other is a control line. We must understand that control line must be present in the testing kit. It is how the testing kit is designed. Whereas the test line can be present, cannot be present, depending upon whether the person is COVID positive or COVID negative. Suppose the person is COVID positive, there are antibodies on this line. The antigen, the foreign material, is taken from your saliva or the swab. It is kept here and then it transmits to the test line, and test line will show a result if you are positive. Say if you are not positive, that means you are COVID negative. There is no test line. However, if the control line is absent by any chance, that means the test is invalid. You have to take a new test again, or you have to go for RT-PCR. Before ending this video, I would like to convey that this rat testing kit is a point of care diagnosis, yes, but the tests are to be confirmed using an RT-PCR again. And that is the reason why we do not have most rat kits. Rat kits are generally provided to regions where RT-PCR can't reach. So, after explaining all of these things, I would like to leave you with 3 questions. The first question is, consider the following statements. Antigen is a component of the pathogen that enters the human body. Pathogen means virus, bacteria or fungi, or as I have said, any foreign body that is not your own pathogen. Antibody is produced by human immune system to defend against the pathogen. Among these 4, find out what is the right answer. Next is PCR. Where is PCR used? We have studied about PCR usages in SARS-CoV-2 diagnosis, which is COVID diagnosis. However, PCR can also be used in other things. Go do some homework and find out where the PCR works. Next is enzyme that converts RNA to DNA. I am sure we have discussed this in our video. Try and find out which one is the answers. However, answers will still be given in the description below. Thank you so much. Please subscribe and like our channel. We will be posting more such interesting videos to you. Thank you.